Hi, in this video, I'm going to teach you how to use the website for the makeup lab. So the first thing you're going to notice is that some of these boxes are in yellow. Those are the ones that you need to fill out, as well as these ones here in color. So the first thing you're going to be doing is typing in the specific question you and your group are going to be answering, and then you're going to state the null hypothesis. Now, in this sheet, you have some helpful hints along the way. So for example, the conditions of the experiment here, it tells you that if it does not apply to your experiment, leave it blank, because the website will allow you to change multiple things, but the only things you need to be focusing on are the ones that pertain to your question directly. Make sure that you're reading the background information that's in your notebook to help you with what you need. And then here is where you're going to enter in the values you're going to be using. Again, only the ones that pertain to your experiment. So let's look at the experiment page itself. That is this tab right here. So you can change, as I mentioned, there are multiple things that you can change in this experiment, but make sure you're selecting only the ones that pertain to your question, because your question may be different than other groups. So once you have set up your experiment, and I'm just going to leave this as kind of that's default, you're going to hit run simulation. So once you have selected your variables, make sure you record that on your spreadsheet, and keep in mind that you're only going to be testing one independent variable. So that's the one that you need to be changing, and that's the one you should be recording. So for example, if you were testing temperature, you would change the temperature on here, and then your first temperature you test would be in the red box on this page right here. So that's where you would put your first temperature that you're testing. Then your second temperature testing would go in the orange, your third, your fourth, your fifth, your sixth, your seventh, and make sure you read the background information to find out what that range of temperatures it is. Same thing would happen if you're testing pH. You would put your first pH you're testing here, your second, your third, your fourth, your fifth, or your sixth, your seventh. So when you are going to run your samples, you're going to put your information here. So for example, I would be Lopez, and this is my first trial. So I'm going to put Lopez 1. Okay, and then I'm going to go through and test the different experiments or different independent variables for each of these boxes. Then I'm going to go through and I'm going to do it again. Lopez 2, this is my second trial run. Depending, the number of trials that you need is going to depend on how many students there are present in your group. But then if there's another one, for example, let's say Ramirez is my partner, then I would do Ramirez 1, Ramirez 2, and so on. Okay, so you're going to put that information there, and then you're going to record your trial runs here. Now the trial runs themselves, let me switch over to that screen. Let me see if I can do both of them at the same time. Share. Nope. Okay. Let me just share this one screen. So once you have selected what your first temperature or pH or inhibitor or concentration, whatever it is that you are testing, you're going to hit run simulation. And then when you do that, you get a nice little graph over here. You see the enzymes are moving around. Everything is labeled with what it is here. What you're going to wait for is your information to plateau. In other words, over here on the graph, something is going to stop. And when it stops, you don't have to worry about getting to it right away. It's not going to be a problem, because what's going to happen is that once you have, so in this case, starch is what I'm breaking down. Once you have reached your maximum amount of product, then it's just going to keep running. There's nothing you need to do. So I can see that glucose is the only molecule left in here besides the enzyme. So amylase and maltase are both labeled. And keep in mind, you only want conditions that pertain to your experiment. So you shouldn't have things in here if they are not being directly tested. So then what you can do is you can hit pause. And then if you're worried that you had missed the time, you can hover over any of these and it'll give you the exact time. So it gives it down here at the bottom. It gives you the exact time in which that you had no more substrate left. Okay. And then that's the time that you're going to record, because we're doing reaction rates. Now, one of the things you're going to have to do as well is take a screenshot of each trial you do. So I don't need the data. I need the data to be done here. You can use snipping tools, probably the easiest way. You can also take a screenshot. And then what you're going to do, so I'm going to take a screenshot here. I've now taken a screenshot of my lab setup. Then you're going to include that in your lab report. So over here on the lab report itself, here is student trial 1. So I'm student 1, and this is where I'm going to insert my screenshots. Okay, you may need to resize the window, that's fine. But you're going to have a screenshot of each of your trials. If you do more than 6 trials, then just keep copying them down here at the bottom. But each student needs to turn in your trial. So you'll have student 1, there'll be student 2, student 3, student 4. You can just go to here, go to duplicate. And when you duplicate that, it's going to make a copy. Oops, I should have taken that out before I duplicated it. Change the name of this. You're going to go to File, Rename to the name of the student, whoever's doing these screenshots. When you're done with your screenshots and you've entered in all your data, so I'm just going to make up data here. Okay. And again, I'm just making up data. I don't know what these values are going to be. The last chart over here is going to give you and automatically generate a SEM bar for you. Now, with this SEM bar, or this bar graph, sorry, it's going to depend on the number of trials that you do. And the color of the graph is going to be whichever one you did. So for example, this here, this red one is the red data that I went through or that I typed in over here on this sheet. So be careful, because the SEM bars do not get generated for you automatically. Okay. In order to get the SEM bars, you need to go in and in your notebook, follow the instructions on step 6. So you need to add SEM bars to this data. And so in your notebook, it gives you step by step instructions. So make sure you have SEM bars on each and every trial that you have. Okay, the instructions begin at number 6. Without SEM bars, your data is not going to be valid or useful. I hope this video was helpful. Thanks for watching. Bye.